project on understanding butternut and butternut canker disease to guide restoration.
Thanks, Matt. Can everyone hear me all right? Okay, awesome. So Aziz did a really excellent job describing what we've been doing and the work that he's been leading, in particular with the genotyping over the last several years. And so the project here that was just funded is really kind of the next step, right? So we've screened trees for resistance, over 300 families, in fact, open pollinated, that cover 22 states in the Eastern U.S., so a good swath of the Eastern U.S. has been represented in our study. And through Aziz's work, we've now genotyped the remaining living trees. So we know roughly which trees are resistant or susceptible and whether or not they're butternut or hybrid butternut. And if they're hybrid butternut, what flavor of hybrid butternut they are, as Aziz talked about the differences. So with this project, we wanted to leverage what we've been doing for the last basically 25 years here at the HTIRC, but the efforts actually started even before that in the early 90s with now retired Forest Service scientist from St. Paul, Minnesota, Mike Austry, who initiated some of the first butternut collections in the early 90s. And so that is to take what trees we have identified as being resistant and then outplant them into sites that conceivably could be suitable for restoration and see how does our resistance hold up. If you've been here at Martell and walked around any of our butternut plantings, which if you've been into a previous HTIRC meeting in the past couple of years, we've probably walked out to the butternut, and we have a really heavy disease pressure here at Martell that likely doesn't reflect disease in the real world. Plus, we're planting a lot of susceptible individuals in close proximity, which again is not necessarily reflective of butternut out in the forest. So that's really what this project is seeking to address, is to help identify places where we should consider planting butternut and then see how stable the resistance is in our resistance screening program that we have here at the HTIRC. So don't need to get into this because Aziz actually presented the same exact slide. I guess I should have paid closer attention when he shared his slides with me yesterday. But as he mentioned, hybrids show greater resistance to the canker, but we may have some butternut that have acceptable levels of resistance for restoration. So as Aziz mentioned, we've been rating trees, and so that's actually where we're going to come in with our study. So we took that ratings data and we actually did quantitative genetic analysis to not only identify the most resistant individuals, but the most resistant families. And this was really important to us because we can have quite a bit of variability within families. Some of that may be due to differences in hybridization that occur between families, or for other factors. And it can also be related to site differences. So Aziz mentioned in the spectral work, we have like high disease sites and low disease sites. So in some places where we have less disease, we're going to have more resistance or a higher absence of disease just because we have less disease pressure. That doesn't necessarily mean those trees planted in those sites are more resistant. They just haven't been exposed to the pathogen at the same levels. So we screened over 300 open pollinated families, and like I said, that represents kind of a range-wide representation of material from the Eastern U.S. So kind of the takeaway messages from the initial quantitative genetic analysis that we've done, that was done by Jim Warren, who's a fellow Forest Service colleague based here in West Lafayette, is that resistance is heritable, but it's heritable only at high disease sites, which makes perfect sense. If we don't see disease, we are not going to necessarily see the trait that we're interested in. So just to break down what this figure is, so these are plot 53 and 51, which Aziz talked about. Those are here at Martell. Dark blue is the most susceptible, and yellow is the most resistant. So we have a lot of very susceptible trees here at Martell. But plot 44, which is down at CPAC, it's actually a sister planting of plot 51, so it shares around 150 families, or both planted, the same families are planted. We have way more resistant trees, and that's because we have way less disease. Now, since we first rated these trees, we've had more disease come in, but we're still not at the same levels of disease down at CPAC that we have here at Martell. So we need to continue following those trees into the future in order to get an accurate assessment of susceptibility in that environment. And that's an important note too, is that we can't really just go in at one time point and measure disease. We have to go in at multiple time points and look at changes in disease and changes in tree health over time. So the objectives of this study were, number one, to identify butternut selection, so families that had demonstrated higher levels of disease resistance. And I will say too that I'm using resistance perhaps somewhat liberally because we don't really have clearly define like what's resistant or susceptible. But I'm a plant pathologist, and in my experience working with disease resistance in trees, particularly caused by non-native or invasive pathogens, we often have what's called quantitative resistance. So this means we have a variation in susceptibility. And this is what we have with Butternut Canker. So it's when we say we have a resistant tree, that doesn't mean it doesn't have disease. It just means that it has less disease than less resistant or more susceptible trees. So it's a continuum. So that was our first objective, was to identify the most resistant or least susceptible families. The second was to identify sites that were suitable for restoration. And then finally was to actually do this real-world testing of performance. So are the trees that appear to be the most resistant here in Martell, like does that resistance hold up? And maybe trees that are slightly less resistant, maybe they're more resistant in other environments. Like that's a question that we're interested in asking. So to do the first objective, first we need to identify those selections with the highest levels of resistance. And to do that, we use quantitative genetics. We use two different methods. I won't get into the nitty-gritty details. I'm definitely not a geneticist, although I've learned a lot about genetics over the last few years. And so we use two different approaches to rank families based on susceptibility. So this provided a breeding value. So similar to those index values that Caleb was talking about in his tree improvement talk, we did something similar. We just used different types of statistical approaches to do so. Then, because we do have some differences in how these approaches rank families, we average the rank, and then we use that in combination with information like the genotype, so that was leveraged from the project that Aziz discussed, and also the location of where those families came from, which Lenny asked about, and I have a map here in the next slide or two. So we took that all into consideration in selecting which families we're going to include. So as Aziz highlighted in his presentation, the most resistant families are hybrid. But the good news is that some butternut families are less susceptible. I was actually really excited for a time because I was like, oh my gosh, the most resistant family is a butternut. And then we did more genotyping and discovered it was actually a hybrid. So that humbled me, and luckily Aziz is really good at genotyping and is using the best tools to do it. But anyway, so these are kind of like the top 10-ish families that we selected. So our goal was to include 10 families in our study. And we have selected more families because if you've ever grafted butternut, it's not super easy. And I've never grafted butternut, but I know it's not super easy. So we're trying to hedge our bets, and maybe not all families will take. So we picked a kind of a range of families. So generally speaking, our most resistant families are hybrid butternut. They come from Connecticut, Indiana. But we also have some families that come from West Virginia and Ohio and even North Carolina. So they do kind of center on this like central Midwest. Now that could be due to a lot of different factors. One being a ton of our families are from Indiana and from the Midwest, so we could just have a bias in our families, and that's why we're seeing more resistance. It could also be because those families are better adapted to these environments, and so they're just growing better. And that's actually one of the things we'll be able to address in this study, is actually look at that G by E effect to see how the environment influences resistance. So we did kind of relax some of our criteria. So Basically, we have families that are in the top 5% in terms of resistance, all the way down to around 20 or 30%. And we did that in part 2 to make sure that we're including butternut families. So like our most resistant butternut family is around, so it's like the top 15% of all families. So we screened around 340 families just to give you a perspective, which I think is really encouraging and exciting. And with our back cross, just to note, those we selected specifically trees that were back crossed with primarily butternut DNA. So we do have back cross families that are primarily Japanese walnut, but we decided not to include that. We wanted to focus on those back crosses that are majority butternut for this study. So this map, the blue dots show where our families come from, and the other 3 colors are our proposed planting sites. So we're collaborating with staff from the Northern Research Station to put in a planting nearby the Wayne National Forest on the Benton Furnace Experimental Forest in southeastern Ohio, with John Butner from the Northern Research Station up in Burlington, Vermont. We're planning to work with like a nonprofit organization there. He's working on the logistical details to solidify that planting. And then we're also going to put in a planting at Connecticut Agricultural Experiment Station in collaboration with Susanna Perio, who is a scientist there. So just this week, scion was collected from our selected families. So grafting will begin this spring, and our goal is to plant next spring, but we are planning to graft over multiple years because we may not get the numbers we need at the first go. So kind of just to summarize what we've done thus far. So we finalized the quantitative genetic analysis to identify the most resistant butternut and hybrid butternut families and leverage that ongoing genotyping efforts to confirm the species. We selected families and we're partnering with expert grafters to begin the grafting process. We have partners that are willing to host 3 different plantings. And then we have a collaborator on the project, Carrie Fuhr, who's an assistant professor at Virginia Tech, has a postdoc, Carl Bascom, who's begun a modeling project aimed to integrate the information from our screening program with landscape and demographic data to identify optimal sites for restoration. And also just to understand kind of the relationship between landscape disease dynamics and the susceptibility and hybridity that we see in our screening program. And finally, we recruited a master's student to begin work on the project starting in the fall. And with that, I'd like to thank all of the collaborators on the project, and if there's time, I'd love to take a question. Thank you.